Good morning and welcome to this BitPlane hosted webinar. My name is Lindsay Hamilton. I am the West Coast representative for BitPlane. BitPlane is a developer of Imaris, a software solution specifically developed for the visualization and analysis of multidimensional microscopy data. During this webinar, we will be hearing a talk from our guest speaker, Dr. Danfeng Chai from the National Institutes of Health on modeling and analysis of collective cell migration. Immediately following Dr. Chai's talk, I will be introducing the new analysis tools available in Imaris that specifically cater to developmental and cell biology research. There will be time for questions directly after each talk, so please type any questions you may have into the chat window. This webinar will be recorded and sent out to all registrants. It will also be available on our website, www.bitplane.com/learning. It is my pleasure to introduce our first speaker. Dr. Danfeng Chai is currently a Damon Runyon Cancer Research Foundation funded postdoctoral fellow in the Jennifer Lippincott Schwartz Lab at the National Institutes of Health. She is interested in understanding how cells interact with the environment and each other, and how these interactions lead to changes in cell behavior. She received her PhD from Johns Hopkins University School of Medicine in the lab of Dr. Denise Montel in 2014. She moved to the University of California, Santa Barbara in the Montel Lab as a research associate before moving back to the East Coast. In this webinar, Dani will provide insights into the research she performed in Denise Montel's lab on how combining live cell imaging and theoretical modeling reveal the microenvironment's influence on optimum size of a migrating cell group. And with that, I'd like to pass it over to Dr. Chai.
Hi, everyone. Thank you, Lindsay, for the nice introduction. Welcome to my webinar. And so today, I want to focus not on the details of modeling, but instead on the revelations we've got by combining the live 3D imaging of this beautiful Drosophila ovary, as you can see here, with mathematical modeling. And I hope my talk will be not only helpful to people interested in cell migration as this general topic, but also the general public who are interested in knowing more about this disease processes such as cancer cell biology. During this process, I will also touch on how Imaris will help us cell biologists visualize this process of 3D cell migration and also draw quantitative information out of our data, which is important if we want to publish really nice paper. So as Lindsay mentioned, in Denise Montel lab, we're interested in studying cell migration. So why do we care about it? Because, for example, cell migration is heavily involved in the developmental process. And as you can see here, this is a cross-section of the vertebrate spinal cord. And showing green is a single axon, which is able to migrate long distances and following multiple cues until they reach their final destination. So cell migration is an indispensable process during cell development, development of the body. Not only that, cell migration is also heavily involved in the disease processes. For example, if we found the primary tumor developing in any of the organs inside of a patient, it's a bad thing. But this primary tumor don't become deadly until cells start migrating away from this tumor and form the distant metastasis in the distant organ. So in this process called cancer cell metastasis, cancer cells travel in the tissue into either a blood vessel or lymphatic vessel, and then also travel out. So cell migration really plays an important part in a cancer cell metastasis, this deadly process. And targeting them, understanding cell migration is important for targeting cancer and also understanding many other important and deadly diseases. So as the examples here shown, cell migration are usually shown as single cells traveling alone, long distances. But actually, as now we're able to do live imaging in the live samples in the living organism, we're able to see increasingly that morphogenetic movements are really diverse. And many cells travel while maintaining a group together, and they're adhered to each other. For example, this is a movie showing you the tracheal development in the fly embryo, which is similar to the lung development in mammals. And those cells maintain their adhesion to each other. And the second movie shows you the mammary gland development in the mouse, and cells also adhere to each other when they travel. And the third movie is the Drosophila dorsal closure, which is similar to the wound closure in the mammals. And the last movie is the zebrafish lateral line development in the zebrafish embryonic development. And lateral line is the very important sensory organ. So these cells all like maintain group adhesion when they migrate. So not only that, we can also see that cells travel in diverse environments. So the first image shows you that this is an example of cells traveling in 2D on the extracellular matrix. So this is usually what people are seeing when they plate the cells on the flat cell culture dish. But not only that, cells also travel in our body on top of other cells in a 3D environment. And this is an example of the neutrophils going out of the blood vessel and attacking the bacteria that's causing this inflammation. So in fact, as patient samples become more available from cancer patients, people increasingly found that in this real environment, in this 3D environment, cancer cells tend to form clusters when they move away from the primary tumor. As you can see here, stained with actin, the cells maintain their adhesion to each other when they migrate out. So this really raises the question of, so this was not really readily seen when you plate the cancer cells on a 2D culture dish. So this really raises the question of whether this 3D environment, this extra dimension requires that the cancer cells or any migrating cell to maintain a cluster. And does this maintaining this cluster give the cells a relative advantage in doing cell migration? So this is what our research here tried to find out, and we had some answer to this question. And before we go to that, a brief introduction to the direction sensing in cells. So this is a single cell, and when you play this cell in the extracellular chemical signal, which instruct them where to migrate, the area where it's where of the cell where it's facing more chemical signal will be more activated, and the other area facing the shallower the less signal will be less activated. So the first part is signal sensing in different parts of the cell. And after that, intracellularly, there will be a signal amplification process. So this front of this cell will be more front and back will be more back. And then the cell here become polarized in this gradient, so that this become dramatically, as you can see, elongated, so that the front of the cell have more, for example, actin machinery that's important for sending out protrusions and migrate. In the back of the cell, maybe having more myosin that's able to contract the back so that they can move away from where they are. And this is the basic principle of cell direction sensing in this gradient of chemical signals. So this is in the example of single cells. So in the collected cell migration model, this is more complicated. But we hypothesize that since when cells form a cluster, they are much larger than the single cell here. So the front of the one side of the cluster versus the other cluster, side of cluster will have a different, much larger difference in reading reading this signal, so that they will have much more different response. So they, as a whole cluster, they might be further polarized and able to move better in this shallow extracellular gradient. But this is just a hypothesis, and this is what we try to test in our research. So as I briefly mentioned to you before, we use this very nice Drosophila ovary model, the border cell migration model, to study collected cell migration in a three-dimensional environment. So as you can see, this is a female fruit fly, and we can culture them in the lab. And if you take out a pair of ovaries here, and each ovary is made up of many strands of ovarioles. And if you take out this single strand of ovariole and look at it under a microscope here, you can see it's a nice assembly line of egg chambers, all the way from the stem cell stage, and they undergo multiple stages of development until they become a mature egg that could be laid later. So border cells are this group of cells which are formed early at stage 8. And as shown in green here are a pair of polar cells. And they're able to secrete the JAK-STAT signaling to recruit the neighboring epithelial cells, shown in red here. And those cells become migratory, and together with polar cells, they can move in between the nerve cells until they reach the oocyte. So this is a really traceable collected cell migration model that happens every time at stage 9. And this is to show you in more detail of this stage 9 egg chamber. So this is surrounded by one single layer of epithelial cells. And so, but the whole egg chamber is encased by an extracellular matrix not shown here. Inside these epithelial cells, there are 16 germ cells. The big fat one at this posterior end is the oocyte, which will later become a will become part of the mature egg. The other 15 of them are germ cells, and those germ cells are called nerve cells, which can provide nourishment to this oocyte. So this border cell cluster here, which are formed by polar cells and the migratory border cells surrounding them, they they delaminate from this epithelial layer, move in between the nerve cells until they reach the oocyte. So as a cell biologist, we like to visualize things. And Imaris really come into play in this process because I'm sure Lindsay will be able to show you in more detail later, but as you can see, the border cell here, they send out protrusions, and this red path here is the junctions interface between those big fat nerve cells, and their nuclei were stained in blue here. And the path is in between those nerve cells, and they will avoid those the ring canals, and then until they reach the oocyte. So this is really a 3D environment that they are migrating through. So in our lab, we have figured out the secret ingredients to really keep this egg chamber happy and image this whole process live. And we label the border cells with green and nuclei with red. And you can see this process takes place for about 3 to 4 hours, and they just reach their destination. And since this is live imaging, there are a lot of information we can extract from this data. And so this comes in place the modeling because we, as I mentioned to you before, we try to understand if cluster size really matters, if forming a larger cluster really helps the cells to migrate better in this complex 3D environment. So we want to build a model to see what's the relationship between the radius of the cluster and the cluster migration speed. So as shown here, we simplify the border cell cluster as a sphere, and each cell is able to experience the extracellular chemical gradient and then elicit a response function. So this function is shown in here, and so the Cx is the concentration of the chemical gradient at this specific X location. And epsilon is the ratio between the receptor of that chemical signal binding divided by endocytosis rate. So this is a nice response function, and we think that the force that each cell experiences is proportional to this response function related to chemical signal binding and reception. So if we integrate the Fx that's experienced by every cell in this cluster together, we can get this complicated total force Fx at this X position experienced by all the cells. So if we plug in the concentration of the ligand, and we can in fact do some calculation and find out that its Fx is proportional to R to the third. And it's also intuitive because this receptor binding of the chemical ligands were proportional to the surface area of the cluster, which is R squared. And then the larger the cluster is, the more different the front versus the back of the cluster is. So there's another R. So together there's R to the third. But if you want to know more details of modeling, you can go back to our PNAS paper. So according to the Langevin equation, Vx, the velocity of this cluster at this time point here is equals to Fx total force divided to the friction coefficient, lambda here. And so when we try to think of where the friction comes from, we think that in our system, there might be two components. The first component is the lambda adhesion, where we know that there are adhesion molecules between the border cells in nerve cells, so that it is proportional to the total surface area because the receptors, the adhesion molecules are on the surface of the cluster. So it's proportional to R to the square. And also, we think that because it's a 3D migration and they are migrating in between the big fat nerve cells, and there are a lot of cytoplasmic components in the nerve cells. So there's viscous drag that's being experienced by the cluster, which can also slow it down. And this viscous drag, the lambda vis here, according to Stokes' equation, is proportional to R. So we don't know the relative contribution of these two lambda here. But later we found out that actually the viscous drag when at a larger R is really dominant when the cluster become bigger. So this movie I've shown you before is a wild type border cell migration movie. Since in a wild type situation, there can be varying number of how many border cells there are in the cluster. So we can extract this information by looking at a bunch of wild type movies and then get the cluster diameter and velocity. So how we do that is to use the surface tracking of Imaris. And then after we track the surface, we're able to get the statistics, and from the bounding box algorithm, we can get the real-time cluster diameter. And then also from the statistics, we can get the real-time speed of each time point. So this has been really helpful to us. But we know that for the wild type, there's really still a limited range of the cluster diameter. So we try to go ahead and generate smaller clusters and also larger than usual clusters. And then with these three data sets, we try to plot this speed and radius plot. So this is showing you how we generate smaller than usual clusters. And we know that from our previous paper, the ECAT herring is an adhesion molecule that binds the whole cluster together. And this is the immunostaining, and this higher, the redder means higher concentration. So you see that in the central pair of polar cells, there are a lot of ECAT herring. So if we knock down the ECAT herring specifically in this central polar cells with our powerful fly genetics, we are able to break down this cluster. And as you can see here, you can see there are two clusters that we knock down ECAT herring in the polar cells, and there are two different clusters in another situation. And you can also generate maybe three clusters. So this is how we generate smaller than usual clusters, and then we took movie out of this mutant cell migration process, and you can see that the cell clusters are smaller than the wild type, but we're still still able to trace their migration speed, and also calculate how large each cluster is. So let's finish the movie. So as I said, we're also able to generate the extra large cluster showing here. We can do that by generating extra polar cells, so that those we can also take movies out of it and see how well they migrate. So this is what we got. And you can see that when at this smaller cluster diameter, this velocity of the cell of the cluster really increases linearly with cluster diameter. And up until a point that where increasing cluster diameter no longer gives the cluster advantage. At larger than 24, they kind of level off. But then when you have a even larger cluster, the increase in velocity, this advantage no longer is here. So this is really interesting results, because this shows us at smaller diameter, this larger increasing in larger cluster size really gives the cells an advantage in navigating in the 3D environment, but until a point that this really no longer helps. So what we think is happening here, as I shown, is what we as shown in modeling, that the viscous drag where this cluster here is experiencing are from the surrounding nerve cells. And also, so this egg chamber is also surrounded by a tight extracellular matrix. So really, this migrating cluster are experiencing a lot of extra viscous drag. But when they're smaller, they don't see that much, and they are able to gain this advantage by increasing in size. But when they further increase in size, so this advantage no longer exists. So that's why in our model, we don't see, even though the increase in size helps them to migrate, but we don't see really, really super large clusters. So So this result is really interesting comparing with cells traveling in the 3D in a 2D environment, which is a case where people usually look at cell migration, where they plate the cells on the cell culture dish, because this size speed correlation was not reported before. As you can see here, so this response function and force are still here, but since we have the one less dimension, the force now, total force now is corresponds to r square. And here the Vx, the velocity, is equals to Fx divided by lambda, but this lambda no longer have this component of the viscous drag because when the cells are in the culture dish, this fluid on top of them are considered infinite, and they are not surrounded by the extracellular matrix and not in the 3D, so that they do not have a large influence on the migration, a negative influence on the migration of the cluster. So this component is no longer here, and the only friction coefficient component is now the adhesion, which is still proportional to r to the square, r squared. And so now if you divide fx by lambda, then Vx is no longer related to r because they just cancel out. So I think that's why looking at cells traveling in 3D environment is really important. So to conclude, we found out that in the 3D environment where a lot of cell migration naturally occur, larger clusters move faster. And then this faster gaining speed happens until the cluster reaches certain size, where this viscous drag from the surrounding tissue will dominate and no longer give them an advantage in cell migration. While in 2D, speed is not correlated to cluster size. So this insight we're really we're very excited to get. And another important question we want to answer is, what is the chemical gradients that cells experience look like? Because according to the traditional view, that's what people usually draw. People think that the generation of chemical gradient would consist of a source, the cells where the source is where there are a group of cells that constantly secrete those chemical signals. And this signal is, on the other end, depleted by a group of cells which acting as sink. So they endocytosis those chemical signals. So with the collaborative action of source and sink, we get a gradient of chemical signals. And the migrating cells kind of migrate in this huge empty space so that they're able to sense differently the concentration of the chemical gradient from the front to back and able to polarize and migrate directionally. So of course, this is far from what was happening really in vivo in the living organism. So because we know that cells don't travel in void space, they travel maybe on matrix or on top of or in between other cells. And so it's really hard to know like really what the shape of the chemical gradient looks like. And also the cells experience, they will usually integrate multiple signals when they are migrating. So previously, people trying to understand and study the directional migration in response to gradients by really generating them artificially. And this has revealed many important insights, actually. The first example is the generating of a gradient by our agarose gel here, as shown here, and this chemical attractant was able to diffuse into this gel so that the neutrophils here contained here will be able to move through this gel and reach this chemical attraction. And this is showing you the generation of chemical gradient by microfluidic devices. So, I mean, these are all nice, but it's what was really happening in vivo and in the living organism was not really known. So luckily, we have mathematical modeling to help us find it out. And we can use the same migration speed information to deduce what really the chemical gradient looks like. So as you can see here, to remind you, this is a Drosophila egg chamber, and the border cells here are really guided by multiple signals when they are doing directional migration to the oocyte. The one signal is called PVF1, and this is actually PDGF and VEGF related factor. And people studying cancer metastasis or angiogenesis are familiar with VEGF. So these are really homologs to what's in the mammals. And when you stain that, the PVF1, this is the secreted factor, you can see it's highly concentrated at the oocyte, and also the nerve cells will have that. And the border cells, they have this PVF1 receptor called PVR, showing green here. So they read this PVF1 and then migrate directionally. But as you can see here, we're only using the antibody staining. We're only able to reveal the bulk of signals. So, but what really matters is only this tiny fraction of signal along the migration path in between the nerve cells that those border cells sense. But in antibody staining, we're not able to really distinguish them. And so the other factors that border cells experience are called Karen and Spitz. And those signals, as you can see here, are also concentrated at the oocyte and diffused out to the nerve cells, but though they signal to the EGF receptor on the border cells. So you can also see that you don't see those by antibody staining or other methods, you don't see this gradient. And this is what we try to find out by mathematical modeling. And so this is the movie showing you that if you knock out both direction sensing receptors, the cells don't migrate directionally. They will send out protrusions everywhere, and they don't move to where they should be, where is this giant oocyte here. So from the response function and the total force we got before, we see that if this chemical gradient follows a linear increase, then this force, total force here, actually will decrease along the path of migration, along this path of migration. So that because this is easy to imagine, because when you are further along in the migration path, the receptors will become saturated and then you no longer tell the difference between the front of the cluster versus the back of the cluster. So the force, total force decreases along migration path, and this Vx, which is proportional to F, will also decrease. So different colors here just show you different values that can be plotted in this, but they all show a decreased trend. But surprisingly, if we use the exponential chemical gradient to plug in this force, a total force function, we see that force no longer monotonously decrease. It will increase first and then decrease. So that there will be a higher point of velocity along the migration path, and then it will decrease. So this is what the difference that a linear chemical gradient versus the exponential chemical gradient will make. And so we plotted many different migration paths and with the wild type migration profile. And you can see that from this follows this overall decreases this when they are towards the end of migration, the one here. But you can see that there's an initial increase in speed and then a decrease in speed. So likely in the wild type cluster, there is they experience the overall combined exponential chemical gradient. So we want to know, like, because we know there are at least two receptors, the PVR and EGFR, and they have different sets of chemical signals. So we want to see the relative contributions of these two signaling pathway that leads to direction sensing. So we use the dominant negative form of EGFR to knock down this EGF receptor in the border cells, so that only the PVF signaling will be present here. You can see that there is still an initial increase in speed and then a decrease. So the PVF signaling here, the PVF chemical gradient also follows a exponential chemical profile. And if we only knock down PVR so that the EGF receptor signalings are present here, you can see that this response is more uniform across the gradient, so that EGFR signaling alone is not really important. I mean, not like the dominant force in guiding the cell migration, but still you can kind of see in the middle there's an increase in speed and then a decrease towards the end of migration. So it still follows a weak exponential gradient. But interestingly, you can combine the two parts together, and you can see that you can get a wild type profile as we seen before. So in conclusion, that in the second part, we talk about the chemical signals for border cell migration, which is really hard to measure, but we can deduce it by mathematical modeling, and it shows that it's an exponential gradient. So the implication of this research, which has two parts, is that really it's important to study the cellular processes in their native environment. So if your migration happens in 3D, try to study them in 3D, and find it. So, and because as our model suggests, it really makes a difference when the cells are studied in 2D or in a 3D environment. And also, yeah, so I think that concludes my talk. So I want to acknowledge my collaborators, Dr. Wei Dai, Dr. Mohit Prasad, and Junjie Lo. They helped me with collecting data and also analyzing data. And also Professor Nirgov from the Weizmann Institute, who was the people who developed the mathematical modeling with us. And last but not least, my mentor for PhD, Dr. Denise Montel, who was really kind and nice and in guidance. So this work was done both in Johns Hopkins University and later in University of California in Santa Barbara, and the funding was from National Institute of Health. And if you have further questions, you can email me. I will be answering questions here, but you can later email me at danny.kai@nih.gov. And thank you very much for your attention.
Thank you very much, Danny. That was a wonderful presentation. I would like to open the floor to some questions. So if there are
Mhm.
people in the audience that would like to ask Danny a question regarding her research, please type your question into the chat window. And I will get questions started while we wait potentially for some others. Can you explain a little about how the data or your Drosophila egg chamber data was acquired and some of the problems that you faced while acquiring live cell data?
So basically, we try to make them feel that they're still inside the female fly in the ovary. So we dissect them out, the egg chambers really carefully from the ovary, and which we're really able to do by practicing. And then we culture them in the live imaging media, which we figure out that like insulin is really important ingredient because it kind of signals the fat state of the fly so that they can start making eggs and reproduce. And later on, and we just put them in this culture dish, which will have this membrane that allows the exchange of oxygen and CO2 so that they're happy. Maybe one thing we want to is hard is because we don't want to squeeze, because we have a cover slip and we don't want to squeeze down too much on the egg chamber so that they're still happy. But also, if we don't squeeze down, then they kind of move a lot. So I think that's where Imaris will be helpful. I heard there's the reference frame that can really register the egg chamber so that it doesn't in the at processing it doesn't move too much. But we in our paper we also developed some uh, MATLAB codes to register the egg chamber in order to address this question. So yeah, it's doable when you have practicing and yeah.
Okay,
So
thank you so much, Danny. That was wonderful. If anyone does have any questions, I'm sure Danny would be more than happy to address those via email. But for now, we're going to continue on with our second part of this webinar, which is to discuss some of the Imaris features that have been recently implemented for cell and developmental biologists. So thank you so much, Danny.
oh no problem.
Okay. Okay, so for the second part of this presentation, again, my name is Lindsay Hamilton. I'm the West Coast representative for BitPlane. I'm going to be going through some of the new features that have been recently implemented that should hopefully, for those developmental and cell biologists out there, should hopefully assist with your analysis research. I do also want to point out that my colleague Arvon Tully, who is one of our support team members, will be addressing questions. So if you have questions regarding Imaris or regarding the analysis that I will be discussing and presenting, please feel free to enter your questions into the chat box and Arvon will address them as we go. So to get started, I do want to provide a couple of slides that go through some of the new functionality, and then I'll finish with a couple of demonstrations of some of the new features, specifically with some egg chamber data that was very kindly provided by Danny Chai for the purpose of this demonstration. So to get started, I wanted to mention that we have uh, obviously a market-leading application that's specifically designed for the analysis and the visualization of big data. So you'll see that we have a data set on the right-hand side of an egg chamber that has been reconstructed in Imaris, along with some of the modeling that we utilize for measurement and visualization purposes. One of the issues that people are coming across these days is in order to image these live cell or these compartments, it does take a huge amount of time and storage in order to collect the data in order to generate the analysis. So I'm going to be discussing a few items in regards to handling big data. I'm also going to go through some object detection techniques specifically for embryo cells and their organelles, 4D object tracking, 4D drift correction, and a new coordinate system that we have implemented so you can get measurements based on a user-defined coordinate. I do want to point out that for a couple of these sections, we do have new features that are available in Imaris 8.3, which was released this week. Firstly, there is a new membrane detection algorithm. We also have a new cell division tracking algorithm associated with our new membrane detection. We also implemented a reference frame, and that reference frame can be used for the purpose of alignment as well as for measurements of distance and proximity. So, to get things started, we have a zebrafish embryo, I believe, that was collected on a light sheet microscope playing in the right-hand corner. And this is just here as an illustration of the size of the data that we as researchers are facing these days. And one thing that I wanted to emphasize is that Imaris is capable of handling these large file sizes. So 10 years ago, a large file was a gigabyte. Nowadays, a large file is in the terabyte range. So just to point out, Imaris reads, loads, and processes data that is significantly larger than the amount of RAM you have available in your computer. We also have a new file converter, which can convert your file formats into our own IMS format substantially faster than before. We support 48 different file formats, so all of those formats can be directly converted into an Imaris IMS file for the purpose of visualization and analysis. And once your files are in an IMS format, they open up in the Imaris application within a matter of seconds. So we have spent some time working on making the handling and the conversion of these huge files more manageable for our users. Second item, embryo cell and organelle detection. So for the Imaris users that are in attendance, for a number of years now, we've had the ability to identify cells based on cytoplasmic and also membrane labeling. The three images we have up here are indicating some cells with a cytoplasmic marker as well as a nuclear marker. Here we have a Drosophila egg chamber where we do actually have a membrane label for, in particular, the nurse cells. And then at the edge here, we actually have a section from a tunicate embryo, noticing that there are no nucleus inside the cells. So up until recently, we only had the option to be able to segment our cells using a cytoplasmic marker or using a membrane label that also had a nuclear marker. So one of our latest implementations is a membrane detection that is specifically for samples like our tunicate embryo here with no nuclear marker. So especially for those people working in the live cell community, that's going to make things easier as far as preparing samples for acquisition, but also for the speed of acquisition, being able to collect one channel instead of multiple would hopefully help you to monitor and analyze at a higher temporal resolution. So just to mention, our new membrane detection is a detection that requires no nucleus within the cell for the purpose of that detection. So if I move one last point, the detections that we apply or the objects that we detect can now be split and also merged. So we now have modification tools for our membrane detection algorithm. So just to show you that same tunicate embryo that we were just looking at, this has been segmented using our new membrane detection algorithm. And this is just a movie to represent that that can be implemented not only in a 3D, but also in a 4D model. Okay, the next component that I wanted to discuss is 4D object tracking. So Imaris has had the ability to track cells over time for a number of years. One aspect that we were not able to cater to until December of last year was the ability to track cell division. Now, in our release in December, which is Imaris 8.2, we launched our lineage module, which provides the ability to detect cell division and also generate new plots. So this morning, I also want to mention that a new feature that we have just implemented in 8.3 is the ability to also track cell division with a membrane label. So we have two images here. You'll notice that the image up top has a membrane label. The image down below, which is C. elegans embryo, has a nuclear marker. So in this case, we use the cell module to be able to detect the cell membrane, and below we actually used a spot detection to detect the cell nucleus. So if I just click, we should see a movie of the division of those cells over time. The color coding that I have applied to this is actually indicating the parent of the cells that we're seeing divided. So we have a blue color that was for the original parent that was labeled blue, and then yellow was for the other parent. So not only can we track these over time, but we can also calculate statistics such as generations, division rate, things like that. Now, as I said, we also have the ability to do this with spots and also with surfaces. So I'm going to click to show you this new feature that was launched a little earlier, where we can not only detect where each of the spots are, but also detect the division between two spots. These particular spots have been labeled so that we can see the fate of the cells that were in the original time point. So this makes it easier to visualize those divisions and also to see where those cells end up. Just to also point out, we have 6 different algorithms that have all been improved as far as speed, but these 6 different algorithms give the users the ability to choose the algorithm that best suits the structures of interest that they're tracking. Last but not least, in our December release of Imaris 8.2, we also implemented lineage plots. So this is now part of our track editor, and you'll see that this is a lineage plot for the data set at the top. And you'll see that the labeling that we've applied for each of the tracked cell divisions is also apparent in the lineage plot. And we also have another one here showing you the labeling being applied to each of those parent cells and the resultant daughter cells. Okay. So we also have implemented some changes to 4D drift correction, and that is with the implementation of what we call a reference frame. So here is a little indication of what this tool looks like. We'll be looking at this shortly within Imaris. The green, the red, and the blue arrows are indicating the X, Y, and Z coordinates or axes, and this can be placed in any location within a 3D data set. It can also be changed and modified between time points in a 4D data set. So this happens to be some data that Danny kindly provided for the purpose of this demonstration. So this actually shows here the egg chamber data set that was included in her presentation. And in this data set, we see the outline of the egg chamber as well as the border cells. And in this image here, you'll see that I have applied what we call our reference frame to the anterior part of the egg chamber, and this has been implemented for every single time point so that we can do one of two things. We can align the data based on that reference frame, but we can also make measurements from that point. So I'm going to hit my cursor, and here we go. So we have the egg chamber now playing over time. We can see the border cell as well as the actual chamber itself moving over time. And you'll see that this reference frame has been placed so that it is mapping not only where the anterior part of the chamber is, but also the posterior end of that. So utilizing this reference frame, we can now align the data. So this next movie is actually illustrating the fact that we can orient the visualization of the data based on that reference frame and use what's called a reference frame play mode, which allows you to fix your reference frame and see the alignment of the data. So I'm going to go ahead and press my key to start the movie, and you should hopefully see that the data set itself is actually moving, but the reference frame is staying still. So instead of having a data set or a chamber that's moving all over the place, it's now staying in one place, which makes it substantially easier for measurements and analysis to be performed on this. Now, I mentioned before that we have this new coordinate system, and this is the reference frame that I've just mentioned. However, the reference frame doesn't necessarily have to be used in conjunction with 4D or multiple time point data. In this case, this is a 3D reconstruction of an egg chamber. The difference being that there are actual markers in there for the nurse cells and also the nuclei in the endothelium, and as well as the germ or nurse cells. So in this particular example, and hopefully you can see, we can place a reference frame at a particular point within that embryo, and then all of the other compartments that we have modeled using, in this case, a surface model, have been color-coded based on their distance from this particular point. So we have a calibration down here showing you that 0 is basically a 0 distance, and 300 in red is indicating the furthest distance for that embryo. So this can be implemented, as I said, on single fixed samples, or it can also be implemented with time lapse. So I'm just going to show you a quick movie. Again, the same egg chamber with a reference point that is adjusted for each time point, and the spots in the image represent the nuclei within the epithelial layer, as well as the border cells in the center here. So over time, we can actually give you the distances from this particular origin, and that may be useful for your tracking analysis. Okay, so just to finish up this webinar, I'm going to be focusing on two data sets in Imaris itself. I'm going to be looking at some 4D drift correction, and I'll also be looking at placing reference points in order to get distance information. So I'm going to switch over into Imaris. Okay. So to start off with, I have the egg chamber data that provided by Danny that I would like to show you how we can implement a reference frame. And once I show you how we can actually add reference frames and manipulate those reference frames through time, I do want to show you how we can pull out the statistics or the new statistics that are now being calculated based on that reference frame. So this image we saw recently in my presentation, but also in Danny's, and you'll see that one of the major issues associated with imaging live cells, especially embryos like this, is that there is movement associated with these cells. Danny mentioned that previously she'd used some algorithms to adjust for this, but I'm going to show now the reference frame and how this can be used to fix this issue. So I'm going to go ahead and pause and go to the very first time frame. Now, the reference frame is available in our scene tool list. So at the very top, it is the cursor with the red, green, and blue arrow. And if you place your cursor on there, it will actually allow you to, or show you that it is adding a new reference frame. So if I click on that, it will add a reference frame into the bottom corner. So your image origin, your 000XYZ. And what I'm going to do is I'm going to move that particular origin to the point at which I would like to use as a reference, which is the very top of this egg chamber. So once we're in the reference frame, we can hold down the control key and we can right click on the reference frame, and that will allow us to do a translational adjustment on the position of the reference frame. So this will allow you to move it in X, Y, and also in the Z axis. In this particular case, I'm going to place that at the top of the embryo. And you may notice it's very small in the bottom here. We do have a little green mark, which is an indication that we have placed a reference frame in a particular time point. And the green indicates that this is a translational adjustment or a point. So now I do actually want to show you that we have rotational control for this as well. And to make it a little bit easier for us to adjust based on the orientation of the chamber, I'm going to implement a couple of other features. So we have the ability to make particular planes visible, and we also have the ability to fix particular planes. So in this case, I'm going to fix my XY, but I'm going to make my XZ visible. So this is going to allow me to see and visualize and also to line up where the reference frame orientation will be. So if I hold down the control key on my keyboard and left click, it will allow me to adjust and rotate my reference frame. And just to make it a little bit easier to see, I'm also going to bring up some contrast so you can see the boundary of the egg chamber a little clearer. So in this example, you'll see that by left clicking, holding down the control key, I can move the orientation of that, the rotation of that reference frame. So now that we've done this, you'll notice that if I step to the next time point, there may be some adjustments required elsewhere. So simply by either reorienting or replacing the reference frame, and then in this case I'm going to rotate slightly, it will place another marker or keyframe for your reference frame, and it will interpolate that movement in between the 2 points at which you've changed. So this can be done throughout the experiment, so you can continue to apply both translational and, if necessary, rotational adjustment for the sample of interest, and it will continue to mark those for you. So I've actually implemented one already, and you'll see that I have a series of both translational and rotational adjustments that have been made throughout this time series. And if I hit the play button, you'll see that this reference frame will actually stay at the top of the chamber and represent the orientation that I'd set. Now at any point, and this again is part of the reference frame option, we have a small reference frame icon in the bottom left-hand corner. If you click on that icon, it will allow you to play the movie with a fixed reference frame. So we can reorient the data in whichever direction we would like, and hit the play button. And now you'll see that the boundary of the image will change slightly as we are now fixed to this particular reference frame. I'm going to adjust the contrast a little bit so we can get rid of the edge of the image. And you can see that now we have minimized the amount of movement by that egg chamber. And more importantly, now that we have a reference at the anterior end of this chamber, we can make measurements. In this case, I'm going to show that we can make measurements on the cluster that Danny was mentioning earlier. The cluster bounding box measurement that she mentioned is something that would be of interest. So we can study the longest principal axis and potentially the shortest principal axis of the cluster as it moves through time. So I'm going to go ahead and pause and move back to the very first frame. And here I have used a surface model to define or segment the border cells and also to track them over time. So you'll see that we have a green surface segmentation for those border cells, and we also have a line which is effectively the trajectory of that border cell cluster. over time as well. So just to show you without the playback mode, if I was to go ahead and play that, you'll notice that the whole chamber is moving. So it makes it a little bit more difficult for us to follow what is going on with that border cell or the movements that are associated with it. But at any point, I can click to implement that again. I'm not sure if you noticed, but we do also correct the tracks in order to implement the reference frame. So all of the statistics that are associated with this are in the surface object. And one thing that I was going to show in here is in our statistics tab, we do now have some additional measurements. In particular, you'll see that these measurements will provide the word reference frame, so you know that they are based on the reference frame that you have placed. So in this case, we now have distance from origin of the reference frame as well as the dataset origin, but we can now also give you tracking statistics that are based on that reference frame. So we have displacement as well as position and speed that will enable you to get that data based on your reference. So as an example, I'm going to use our color tab here to statistically color code the surfaces or the clusters based on a statistic, which is distance from origin for our reference frame. So I'm going to do it on the original reference frame, and I'm going to bring this back to the start. So hopefully you can see the purple. I'll move this up a little bit to show this a little clearer. The purple is representing the shorter distance and the red is showing the longer distance. I'll also make this a little larger. So in this case, the distance starts off around 30 microns and goes to about 150. So if I go ahead and play, you'll see that that cluster of cells will actually change in color, representing the fact that that cluster is changing in its proximity to our reference frame or point of interest. Okay, so one other example that I wanted to show you is associated with, again, egg chamber data, but this is a single time point. And I'm going to open up the data set that we looked at a little bit earlier. This is a 3-channel data set. It is fixed, and it does allow us to see the boundaries of the nurse cells and the different compartments within the egg chamber. If I go into the slice view, you'll see that there is quite a bit of complexity as far as the organization of all of these cells within this egg chamber. And here are the border cell cluster that we heard Danny discussing earlier, as well as the nurse cells with their nuclei in this compartment here. So if I go back to the 3D view, I do want to show that a reference frame can also be placed on any particular point of interest. I chose to place this one on the very top of the egg chamber. And just like we saw before, it is very straightforward to reorient and change that particular reference frame using our ability to visualize cross sections of either XY, XZ, or YZ, and also particular planes can be fixed in order to make it easier for the placement. So once a reference frame has been placed, we can implement some of the modeling that we apply in Imaris to be able to extract proximity and distance information. So I'm going to turn the reference frame off, and you'll notice that I have in here the border cells cluster, but I can also add in some of the other segmentation that I've applied to other compartments within the egg chamber. So first of all, I'm going to add in the nurse cells, and also I'm going to add in the posterior and anterior nuclei to show this color coding. Again, the color coding is related to distance and has been set based on the distance from the reference point. If I turn the volume off, you'll be able to see that a little bit clearer, that color coding is now showing you how close each of these structures are. This information can be extracted directly from our statistics so that you can make some particular measurements on structures of interest. I do also want to show you that we do, using our membrane detection in the cell module, have the ability to segment out, in this case, the germ cells or the nurse cells that we also have in the egg chamber. And in this particular case, the nurse cells can also be color-coded based on proximity, but they can also be color-coded on any statistic that we calculate, whether that be the nuclear to cytoplasmic ratio or the volume or the surface area of each of these compartments. And using something like a clipping plane allows us to be able to cut through and slice to see where particular structures are in relation to one another, especially with a 3D reconstruction like this. Now, one of the newer features that has also been incorporated into Imaris in the latest release is the ability to look at what we call a slicer display for a membrane. So if I click on the cell object for the nurse cells, I'm going to go to the visualization tool and I'm going to click on the slicer display. Now, I'm going to just click and drag to move the slice, and hopefully I don't do that too quickly, but hopefully you can see that now in order to be able to see what's going on within a structure, we now allow you to visualize the membranes with a line, and these you can drag using the select mode to different planes within the image. Right now our slicer direction is set to XY, but it can be changed to XZ, YZ, or also an oblique slicer. So this makes it a little bit easier to visualize what's going on. And just as we mentioned before, we can also visualize these border cells. So I'm going to turn off these surfaces and focus in here on the border cells. This border cell cluster can be split into the individual cells that make up this cluster. And just like we saw before, this cluster can also be visualized in the slicer display, again, making it easier for you to see each of the boundaries for the border, for the cells within that cluster. Okay, so that comes to the end. I apologize for running a little longer with this webinar, but hopefully that gives you a sense of what is new in the latest release of Imaris. For those of you that have an Imaris license, we have now made this available in the customer portal for you to download and upgrade. Just to finish off, okay, for any further information on the features that I've discussed this morning in the webinar, you can go to www.bitplane.com/developmental-biology, and that will take you straight to a page that includes all of our new features, as well as the ability to request a demo. I'd like to thank you for your attention today, and hopefully you'll find this useful for your scientific research needs.